Hello guys, today we'll be doing histology of microcirculation. Now, in our body we have blood vessels. These blood vessels have many layers. Some have three, some have one. But the most innermost lining of all of these blood vessels is the endothelial cells, and they're present in all, such as arteries, veins, and capillaries. Now, capillaries are they only have a tunic of media, so they'll only have a single layer of endothelial cells. Now, the structure of the endothelial cell is pretty unique. It has a single layer of squamous cells. It is polygonal in shape. It is smooth, so friction can be reduced. If you view it from a longitudinal section, it has a flat nucleus. If you view it from a transverse section, it has a bulging nucleus. It sits on a thin basement membrane, and the endothelial cell is aligned parallel with the blood flow to obviously reduce friction. Now, what is the point of all of this, these unique structures? It is so it can be related to its function. So, as I said, it had a single layer of squamous cells, meaning that fast transport of oxygen can occur as the diffusion distance is less. And it is smooth, so it can prevent thrombus formation. And it synthesizes substances used in coagulation and inflammation, and it is a selective blood tissue barrier. Now, as we can see here, we have variable palate bodies. Now, these things are small storage granules that are found inside the endothelial. This is the endothelial. And you can see they store two proteins, VWF and P-selectin. Now, VWF is released from these bodies when the endothelial cell is damaged. And the P-selectin, on the other hand, it is released when there is an infection. Now, this is a phospholipid bilayer, meaning that substances cannot just diffuse through it. And for large molecules to pass through, we have these penocytic vessels. Now, how does blood move in the capillaries? Well, capillaries are around 5 to 10 micrometers in diameter. Blood, on the other hand, is 7 micrometers in diameter. Now, RBC membranes are very, very flexible, meaning they can squeeze and they can go through it. So now some capillaries, they can be 5, they can be 6, they can be 7, they can be 8, they can be 9, and they can be 10. So anything ranging from 10 to 7, the RBCs don't need to squeeze through. But anything that's below it, the RBCs had to squeeze through. Now, as you can see in this diagram, the RBCs move in one single line. And you can appreciate the flat nucleus of the endothelium in this longitudinal section as well. Now, this right here is the capillary bed. Now, there are some new structures we haven't seen before, such as the meta-arteriole and the precapillary sphincters. Now, meta-arteriole, they're centrally located arteriole that brings the blood directly from the arterioles to the venules. Look here. While on the other hand, precapillary sphincters, they're found before the capillaries. They're made of smooth muscle cells that can contract and relax, controlling how much blood goes into the capillary from the arteriole. Okay, so now suppose we have our skeletal muscles. Now we're not going to be using them all the time. So now if the skeletal muscles are at rest, we don't want blood to be going through them. So in these cases, our precapillaries they'll contract. These shut the capillaries, and blood will flow directly from our arteriole to our meta-arteriole and to our venule. Now, on the other hand, if you're exercising, we need our skeletal muscles, right? So we need blood to go through it. So, oxygen can go in, ATP can be used, and all of that, and muscle contraction can happen. So we want our precapillaries to relax. Our capillaries will open. Blood will flow in directly from arterioles, meta-arterioles, capillary bed, and to the venules. And now, as you can see in these pictures, when we are exercising, the precapillaries are open, blood goes through it, oxygen exchange happens, and here we close our precapillaries, and you can see no blood goes through it. We just bypass it, arterioles to venules. Now we have lymphatic calories. I mean capillaries. Now our lymphatic vessels, they also have three tunics. And you should also know it's an open system because you can see that lymph goes outside, but blood, like, it stays within the vessels. It'll never go outside. But lymph, it can go into the tissues and all that. So it's a open system. Now, places without lymphatic drainage: the central nervous system, cartilage, bone, teeth, placenta, cornea, and epidermis. Now, why do we not have capillaries in here, lymphatic capillaries specifically. It's because we don't have any capillary bed there. And if we don't have that, then no lymph will be like left. If there's no lymph left, then what's the point of like having capillaries over there? There'll be no fluid to collect, so we don't like need it in any of these places. Now, difference between blood capillaries and lymph capillaries. Blood capillaries are thin one-layered endothelium with a basement membrane. They have a smaller lumen and they have red blood cells. They're more regular shaped, meaning like it's a perfect circle or like an oval. They have marginal folds with tight junctions. They're less permeable as compared to lymphatic capillaries. On the other hand, lymph capillaries, they're thin one-layered endothelium without a basement membrane. They do not have one. They have a thin basement membrane, but these ones do not have it at all. They have a larger lumen with lymph and lymphocytes, and it's an irregular lumen, meaning like it's a very random shape. They have no tight junctions, and they're more permeable as compared to blood capillaries. They can carry whole cells, and that's how cancer spreads in our. Yeah, so here we can see the lymph, very irregular shape, and it got lymph in here. There's one way to also identify this. Sometimes blood can be seen on like these slides, but not all the time. But if you see blood, it's probably going to be a blood vessel, not a lymph. But it should not be like a major way of identifying. But if you do see blood, you should know that. And next, we have a lymphatic system drainage. We have two lymphatic ducts, the thoracic duct and the right lymphatic duct. And the right lymphatic duct, it drains it. What do you call it? It drains the upper part of the head and the right part of the body and the right arm and the right upper thorax, and it drains into the right subclavian vein. While on the other hand, the thoracic duct, it it drains the part of the head, the left arm, the lower like the lower whole part of the body, and it drains into the left subclavian vein. Now how do we classify capillaries? Now our capillaries only have like a tunica media with a single endothelium. So how can you classify them? We can classify them based on we can classify them further into continuous, fenestrated, and sinusoidal. Now let's say continuous capillary right here. Now you can see in the picture, it has a continuous basement membrane right here. It has pericytes. It has these marginal folds. These marginal folds are cytoplasmic flaps, as you can see here, between two endothelial cells. They also have tight junctions between here, and they're the least leaky out of the three. Now where are they found? They're found in the tissue, skin, lungs, connective tissue, cardiac, skeletal, smooth muscles, exocrine gland, and the central nervous system. And it's in the blood-brain barrier because it has less pinocytic vessels. Now, fenestrated capillaries. The basic thing about a fenestrated capillaries is imagine you have a paper, you just poke many holes into them, and you roll it up into a cylinder. That's how fenestrated capillary looks like. Now, the structure of it is that the endothelium, it has endothelium, it has a continuous basement membrane, it has fenestrae, like I said before, right here. These are called fenestrae. They have pinocytic vessels, and like these fenestrae, they have a diaphragm, and it's made of glycocalyx. Now, this prevents large molecules from passing through because like we don't want everything to go through. We only want some specific things. Now the location should be in a place where like excretion takes place. So we have endocrine glands, kidneys, pancreas, and the small intestine villi. Now sinusoidal capillaries. Okay. So the structure of it is a discontinuous endothelial cell and discontinuous basement membrane. You can see here the endothelial cell is discontinuous and the basement membrane is also discontinuous. It has a large diameter and irregular shape. It has many large fenestrae and no diaphragm. It has no parasites, and you can see the nucleus of the endothelium also. Now the location is in the liver, spleen, and the bone marrow. Now parasites. Parasites, they're found with always with mainly with continuous capillaries and the post-capillary venules. They are sometimes found with fenestrate capillaries and they're never found with sinusoidal. Now, what is the function of parasites? You can see here, this is a parasite right here, and a parasite, it can constrict the capillary, so more blood flow can happen. It makes part of blood-brain barrier. This is because it should not be leaky and to prevent unwanted substances. So that's why parasites are there. It can make smooth muscle cells and endothelial cells because it acts like a stem cell, and it can do angiogenesis. And you can see it's also like hugging the capillaries. Now, there are some diseases which can be caused because of parasites. One is diabetic retinopathy and tumor angiogenesis. And the tumor parasite is called hemangioparasitoma. Hem means blood, angio means vessels, parasite means peri means around, and cytoma means cancer. Okay, now post capillary venules. So we first have an arteriole, it becomes a capillary, and then the PCV forms. You can see the capillary. It has a slightly larger lumen, and you can differentiate like that. It has one tunica. It looks like capillary with a larger lumen, as you can see here. It has no analogous artery, meaning that we know that artery and veins have pairs, but there's no pair for the postcapillary venule. And the main function of it is diapedesis of WBCs as an infection. So when we have an infection, we need the white blood cells to come inside our tissues, and it mainly happens in the postcapillary venule. Okay, so here's a question. Organs such as the brain and thymus have a more effective blood barrier because their blood capillaries are of which of the following types? Now, first of all, it can't be discontinuous. We have already stated that sinusoidal, they have discontinuous basement membrane and discontinuous endothelium, and that means it can let a lot of substances pass through. Now, in fenestrated, they have fenestrations. So it does not matter if it has a diaphragm or it doesn't. It lets substances pass through. On the other hand, we have stated that continuous types are the least leaky ones, and they have marginal folds with tight junctions. So A is the most appropriate answer for this question. Okay, this is going to be our last slide. So now we have these large arteries, and they're carrying oxygen. And these are also living cells, and they need oxygen to live. So now oxygen, it can only diffuse a few microns into the wall. It can maybe diffuse through the tunica intima and parts of the tunica media. But this outer tunica media and the tunica externa, they get no oxygen. And when they get no oxygen, they will obviously die. So we need some external source of blood. And that's where vasovasorum comes in from. So now it connects to these outer walls, and oxygen can diffuse through it. And this allows oxygen diffusion, and it can just live. Thank you. And if you have any questions, feel free to message me.